All right, let's jump right in. We are going to break down a really amazing scientific breakthrough, one that tackles one of the single biggest threats to modern medicine, and that's antibiotic resistance. Scientists have figured out what is essentially a genetic undo button for superbugs. And the story of how they found it, well, that's almost as important as the discovery itself. So first, I just want you to take a look at this number, 10 million. What does that even mean? Well, it's a projection, a pretty terrifying one for a crisis that's looming right on the horizon. That's right. If we don't come up with some new solutions, and fast, by the year 2050, more people could die every single year from infections we can't treat than from cancer. Think about that. This isn't a problem we can solve by just, doing more of the same. It requires a completely new, a radical new way of thinking. So let's really frame the problem here. We are talking about the very real, very scary possibility of a post-antibiotic world. A world where something as simple as a scraped knee or a routine surgery could once again become a life or death situation. And look, this isn't some distant, far-off future problem. As you can see from this quote from the actual research paper, it's already here. It's causing over 1 million deaths every year, right now. Bacteria are smart. They evolve, they share these resistance genes with each other like trading cards, and our old drugs, they're just becoming less and less effective. So how in the world do you fight an enemy that is constantly changing and sharing its weapons? Well, you don't just attack it from the outside. You have to get inside the gates. And that's exactly what scientists did. They developed a solution that works kind of like a genetic Trojan horse. Okay, so the core of this whole thing is CRISPR. You've probably heard of it, right? The easiest way to think about it is like a pair of programmable molecular-sized scissors. You can literally tell them exactly where to go in a long strand of DNA, and they'll make a perfect, precise cut right there. It's an unbelievably powerful tool for editing genes. Now, here's the really clever part. Scientists took CRISPR and gave it a new trick. They created this system called ProAge. It uses those scissors not just to cut and destroy the resistance gene, but to cut and then paste in a new piece of code. And that new insert totally breaks the gene. Now notice this. It's not killing the bacteria. It's just disarming it. And this is a perfect illustration of how it works. You see that proamp plasmid on the left? That's like the delivery vehicle. It finds the target bacterium with its resistance gene. That's the AMP-R here. The CRISPR system goes in, makes a cut, and then it pastes in that purple genetic cassette. So what's the result? Well, you see it on the right. The bacterium is still alive and kicking, but its resistance gene is busted. It's been completely neutralized. Okay, but it's one thing to disarm a single bacterium. It's a whole other ballgame to do it for an entire population of trillions. So the next big challenge was, how do you make this cure contagious? So the solution was to package the whole ProAge system onto a special bit of DNA called a conjugative plasmid, and you can basically think of this as a biological file-sharing system. Bacteria are constantly swapping these little plasmids, and so the scientists, they loaded their cure onto a file that the bacteria themselves would want to share with all their neighbors. So clever. And here it is, the delivery vehicle itself. It's a plasmid called pProModV. It's the ProAge system plus all the genetic machinery it needs for that file transfer. So as you can see, the donor bacterium can now link up with the recipient and pass along this entire toolkit, which then turns the recipient into a new donor. It's literally a self-spreading cure. And this is where the story gets really, really interesting. Because when the scientists actually unleashed the system to test it out, it worked. But it didn't just work in the way they expected. They actually stumbled onto an accidental superpower. So they were expecting to see their cut and paste thing disarming the bacteria. And yeah, they saw that. But something else was happening, something much more dramatic. And it was happening way more often. So here it is. This diagram shows the crucial difference. On the left, that's the ProAge event they designed. The purple cassette gets inserted, gene broken. But look at what they discovered on the right. The system was also triggering a massive deletion, using 2 identical bits of DNA as bookends and just poof, snipping out everything in between, including the entire resistance gene. And just look at this breakdown. This wasn't some rare accident. The accidental deletion was actually the main thing happening. A whopping 80% of the time, the system wasn't just disabling the gene, it was completely and precisely removing it. Gone. They had just discovered a brand new, super efficient genetic process. So they gave it a name, homology-based deletion, or HBD. and it basically works like a find and delete command for DNA. The CRISPR system makes the cut, and then the cell's own natural repair machinery, guided by those two identical sequences, just stitches the DNA back together, but without the entire chunk that was in the middle. It is incredibly clean and efficient. Now, when you have a tool this powerful, a totally new question comes up. The scientists realized their accidental discovery wasn't just a stronger weapon. It was potentially the perfect safety switch. I mean, think about it. How do you control a self-spreading genetic tool? What if you need to turn it off? What if you need to reverse it? You need a fail-safe, right? This is one of the biggest challenges in this whole field, and it turns out HBD provided this beautiful, elegant answer. So here's how the undo button actually works. On the left, you see a gene that's been turned off by sticking in that genetic cassette. Now to reverse it, the scientists just introduce a new CRISPR guide that targets the cassette itself. And then the HBD mechanism kicks in, finds the identical DNA on either side of that cassette, and just deletes the insert, perfectly restoring the original gene. It is a true, honest-to-goodness genetic undo button. And how well does this undo button work? Get this. The restoration of the gene's function was nearly 100% efficient. That is an unbelievably precise and reliable way to reverse a genetic modification, which, for the safety of these kinds of technologies, is a massive, massive step forward. So what does this all mean for the future? Well, it means we're not just getting better at writing genetic code, we're getting better at editing it responsibly with built-in controls and, yes, fail-safes. This opens the door to engineering a much safer future. And the potential uses here go way beyond just fighting this one antibiotic resistance gene. Imagine using this to engineer the microbiome in our own guts for better health, or to clean up pollutants in the environment. You could do clinical interventions. You could scrub entire pathogenicity islands. Those are the parts of bacterial DNA that make us sick. Or maybe even reverse a disease's virulence as it evolves in real time. So I guess the ultimate takeaway is this. We're moving from a world of blunt instruments, like broad-spectrum antibiotics, to a world of incredibly precise genetic scalpels. And the final question this leaves us with is, it's a big one. If we can safely and reversibly rewrite the code of bacteria, could we one day program entire ecosystems, from our own bodies to the whole environment, to just be healthier? It's a future that is quickly coming into focus.